right, so let's get into the question about how do we start to calibrate and check our flow cytometry data, which is also going to start showing us, in response to one of those previous questions, some of the useful, free, open software out there that will give you both these capabilities and some nice visualizations to work with. In specific, I'm going to work with one that we recommend from the measurement committee called TASB Flow Analytics. TASB stood for a tool chain for accelerating synthetic biology engineering. This is one that I happen to be involved with the maintenance of, and so it's a favorite of mine and fairly well developed. So let's talk about why we need calibration again. Just a quick review that this gives us great per cell measurements, but the things that actually come out of the flow cytometer are not physical units. Here's a plot of a typical, this graph was taken from FlowJo, for those who know what that is. It's one of the commercial flow cytometry softwares. This is showing what came out of the instrument. It's yellow on the bottom, blue on the side, and when I first started working in synthetic biology and my in the lab colleagues said, okay, here's some great data we got of this inducer, and I said, okay, great. So what? It's yellow and blue. What are the units? And they said, yellow and blue. I said, what? What are the units? Yellow. How many yellows per blue? It depends. I went right back to 3rd grade and they told me that you don't know what a number is unless you know, is it in kilograms or liters or seconds or... So we need to get these into something that has something to do with molecules or something like that. These arbitrary unit outputs depend on your instrument, the configuration of its light power, of its light path, what's going on with other colors, the particular settings that you've got. There's these amplifiers that you can turn up and down. There's the whole thing drifts. So you absolutely have to, if you want to understand anything about what these numbers are telling you, you have to calibrate. But fortunately, this can be done, and it can be pretty easy and pretty cheap. So let's talk about calibrated flow cytometry. So on the right here is a typical calibration path. We start out with some arbitrary unit data on the top. The first thing we want to do is gate it, because we don't care about some of this stuff that's not the things that we want to study. Sometimes you want to study the debris, usually you don't. That gives us filtered arbitrary units. We want to subtract the autofluorescence from those. That's the cells are showing us not just what molecules are in them, but also the cells themselves may be fluorescing. So we typically want to remove that, but not always. If we have more than one fluorescent protein, they may be dumping fluorescence into each other's channels because their spectrums overlap. And so we want to be able to compute what the overlaps are and divide that out. That ends up giving us compensated net arbitrary units. The next thing, again, this is not always necessary, but it's nice to have. If we have blues and yellows or blues and greens or reds and greens, we'd like to be able to compare them. And so when possible, it's nice to have a unit translation from channel to get everything into the same units. And the units we recommend for that when possible are units of equivalent fluorescein. Finally, we can take fluorescein and forward scatter and use some standard materials to relate those to real physical units in terms of molecules and in terms of size and microns. And that ends up giving us calibrated data that we now have something that's standardized, interpretable, readily compared from lab to lab, etc. So to do this, we basically need a few standard materials. There are some wonderful rainbow beads. They're now certified by the US NIST Standards Institute that can give us these equivalent fluorescence molecule units. We can use wild type or null transfected to give us an autofluorescence estimate that we can subtract from the background. Use single positive controls to do the spectral compensation, and multicolor controls to do color comparison. When we do all of these things, we get extremely replicable measurements to about 1.2-fold on 1,000-fold to million-fold range. So this is a fantastically precise measurement. And there are multiple free and open packages which you can use with this. The one that I've got linked down below, TASB Flow Analytics, is one that we support. There's also the Cytoflow package out of the Weiss lab at MIT, and the FlowCal package out of the Tabor lab at Rice. Those were both based off of TASB, and they're close relatives in their capabilities. So let's talk through these specific pieces one by one, starting with gating. This you want to use a negative control for. You want to be often somewhat aggressive in your gating. It's usually better to be a little bit tight than a little bit too loose, especially if you've taken a lot of data. You can afford to throw away some of it. If you've got 100,000 events, you can afford to throw away 20, 30,000. You've still got a lot of events to work with. Better to throw away a few cells than to keep too much trash. What I typically recommend for gating, if you use software like FlowJo, it will nudge you towards sort of a squint and draw a polygon approach, which is very problematic because we're not very good at squinting, and our polygons may not be as good as we want them to be, especially replicable from sample to sample. Instead, I find that it's good to use a Gaussian mixture model, and there's standard methods for that in TASB. So what this is doing, it's looking for basically two log normal distributions, one to find the cells and one to find the non-cell stuff. The second thing that we need to do is, again, use a negative control, typically a wild type, but sometimes a null transfection. That is a transfection with essentially a blank plasmid if you think that your cells may be responding significantly to the transfection itself, to make a model of autofluorescence. And here we'll often see kind of a funny thing. We'll often see that we have events that have negative fluorescence values. Negative fluorescence is not possible. However, what we're not really seeing negative fluorescence here. We're really seeing the noise in the measurement of the instrument itself. So the instrument is giving us these fluorescence values through interpreting a voltage on the detector. And so there's just a little bit of noise in that. And on a range of, say, 10 to the 5th, going up and down a couple hundred is not unusual. But you can see there's a tiny little nudge to the right here and where this is. So we're going to end up subtracting just a couple units. And this also tells us where is the minimum level that we can rely on a number. Once we start getting down into the instrument range, in here, any measurement less than about 50 should be considered suspect, because at that point, we're really getting entangled with instrument noise. The next thing that we need to do is compensate for spectral overlap. And with a flow cytometer, we get to do this in this, what I find, a very beautiful way. You use a strong positive control for each color. So that is a constitutive control. In this case, we've got a constitutive expression of MCATE that's being measured on this Texas red channel. And we're asking how much MCATE is slopping over into our yellow fluorescene measurement channel, which is where we're going to be measuring, in this case, YFP. And this relationship is strictly linear. So what we should end up seeing is that at low levels, it's flat because you're being dominated by autofluorescence and instrument noise. And then as you get to high levels of fluorescence, if there's a significant overlap, it's going to show up as a diagonal line with slope 1 coming up out of that bottom. In this case, we're seeing this diagonal line coming up here, and it's showing that we've got about a little more than a 1 in 1,000 spillover. So this is a really pretty low level. We want to make sure it's less than 10%. I usually recommend keeping to less than 3% if you can. But often you can get combinations like from the fluorescent proteins that the measurement committee recommends that are extremely low indeed. Once you have these models, it turns out that because this is a purely linear relationship, if you make a model of each of these individual pairs, that you get a matrix, which then you can just divide versus the measurements you get, and you unentangle those measurements and are able to compensate. So as long as it's fairly low, like I said, about 3% or less, you can remove pretty much everything quite cleanly. Again, there's standard methods for doing this in the TASB software. So at this point, we've now got the net unentangled measurements for each fluorescent channel. The next thing that we want to do, if we have the opportunity for it, you can do without this, but I really like doing this, especially if you're measuring like the input and output of a device and you want to know how they relate to one another. Then you don't want to get your green units into equivalent fluorescein and your red units into equivalent Texas red, because Texas red is not fluorescein. You really like to get them in the same values. So if you can make a dual control, or perhaps even a triple control in some cases, then you can use this to directly map from, for example, in this case, we're directly mapping how many blues per yellow. And we're finding that the answer is that it's about, so this 10 to the 3 is going to about 2 times 10 to the 3. It's about 2 yellows per blue is the translation here. This is hard to do in bacteria, but if you're working in something like mammalian cells or yeast, where it's easier to disentangle the controls from one another, it's very doable. Last, whether or not we map things to the same value, we can map each fluorescent channel to absolute units. If we can get everything to the same value, it's ideal. Then we can just map FTSE, just i.e. the fluorescein, the green-yellow channel. That's the one we recommend because it's the most common, most standardized channel, into absolute units. There are these wonderful beads that have been developed by a company called Spherotech and now certified by NIST that give you a set of irregular peaks. So if you can see a set of peaks in the intensity spectrum, so this is basically you've got very high peaks and not quite so high peaks and so on, 6 or 8 peaks. If you can see at least 3 of them, then you know exactly which peaks you're looking at. And from that, since they've measured how many molecules of fluorescein each peak is equivalent to, you can figure out exactly what's the mapping between your arbitrary units and fluorescein. And they've done this for a whole bunch of different channels. I recommend running the beads every time that you run a batch because they're pretty cheap and flow cytometers drift some. And these are also fantastic for detecting instrument issues. If something goes wrong with your lasers or your calibration or in the actual instrument itself, it usually shows up as blurry peaks. The result, once you go through all of these, is you get a nice distribution in units of molecules of equivalent fluorescein, much like we were talking about back at the beginning. And this now is something that is highly repeatable, highly transferable from lab to lab, and fantastic for putting into modeling. One last thing, in addition to doing this with the fluorescence, Spherotech also makes a set of differently sized beads that can be used for calibrating forward scatter. This works just about the same way, except that the relationship between forward scatter and cell diameter is not quite linear. We end up doing the fitting a little bit differently. So, in summary, if you're working with flow cytometry data, it needs to be calibrated. This is a multi-stage processing of gating, autofluorescence subtraction, linear transformation, mapping colors to equivalent units, and conversion of equivalent units, and conversion of those equivalent units into equivalent fluorescent molecules via beads with known fluorescence. And we can do the same thing for working with variable size beads to calibrate forward scatter to estimated cell diameter. This may look pretty complicated, but there are free software packages available, including this TASB Flow Analytics, that will give you all of this as a free, open, shake and bake package where you can do it very simply. So I pause here for a moment to take a look at questions. Any questions on these matters before we move on to interpreting our data? All right,